So now that we've established the introduction to protists, we can actually start naming protists, classifying them, and understanding why they're classified the way that they are. Previously, we mentioned the four supergroups of eukaryotes, and we're going to start with the first supergroup that we mentioned, which was Excavata. And we'll entitle this first flowchart on Excavata, Excavata I, and we'll go into the details of what classifies as an excavata and what are some real-life protist examples of these excavata organisms. Now, one thing I want to make sure that we're aware of is that we're studying eukaryotes. Okay, let's not forget that, and we're studying protists within that eukaryotic structure, within that eukaryotic domain, let's say. So don't lose sight of the bigger picture as we go deeper and deeper into these organisms. Remember, we always start at the eukaryotic level. That's why we started with domain eukarya. Now we're studying protists. What type of protists specifically? The excavata protists, which is a supergroup of eukaryotes. So, excavata are going to usually be characterized by the following. So we'll say that excavata are usually, and this is something you'll notice a lot in protist study, that we always say usually or sometimes or some of them, because not everything fits into a nice box of classifying details. Instead, we're going to have some disparities in terms of what fits into what category, but we'll just say usually characterized by, and this is something we'll see a lot also. This term, characterized by, we're going to be seeing this a lot as we study protists, because protists are going to be characterized by certain things. Certain things are going to define them essentially. For excavata, they're usually characterized by these excavated, thus the name of course, these excavated feeding grooves on one side of their body. So feeding grooves on body. And this is specifically on one side of their body. There'll be a figure that I'll refer to that you can truly see this excavated feeding groove. So most of them have this, some of them don't, but let's not get into those details just yet. Now, in terms of studying excavata, you actually have to classify excavata, subclassify them into subdivisions, okay? So now, don't lose me here. We're talking about two different subdivisions now of the excavatas, which are themselves protists, which are themselves eukaryotes, okay? Don't forget that stepwise understanding of where we're classifying organisms. Now, there are three subclades, I would call them, of this type of protist, the excavata. And clades are just another way to classify and group large amounts of organisms. The first one to study are the Diplo Monads. Now these are names that unfortunately need to be memorized, characteristics need to be memorized, but in terms of understanding them, let's remember that there are going to be key characteristics, key characterizations, like I mentioned before, that are really going to drive home certain points about the protists in general, and that's what we're going to be looking for, and that's what I'll highlight especially. For the Diplo Monads, what we want to understand about them is that they have something called a not a mitochondria, but a mitosome. A mitosome. This is a reduced mitochondria, meaning that it's a mitochondria that is not fully functional, that is not the typical mitochondria that we've studied all throughout Biology 115 and even, as I mentioned earlier, in our primary endosymbiosis video. Instead, this reduced mitochondria, it actually lacks a functional ETC. That's why it's reduced. So it lacks functional ETC. If it lacks a functional ETC, you should remember from Bio 1, or at least I hope you can remember from Bio 1, that the ETC was a key aerobic part of cell respiration. This was a part that really absolutely needed oxygen as a terminal electron acceptor. And if you don't have a functional ETC, you definitely then can't use oxygen. And that's exactly the case for the Diplo Monads. They can't use O2, oxygen, to extract energy. So they cannot make ATP utilizing this pathway of the electron transport chain to extract energy, big E for energy. Instead, what they do is they get their energy from anaerobic pathways, and it makes total sense if we understand basic cell metabolism and cell respiration. From anaerobic pathways. If we remember some anaerobic pathways, that would be great. Something like glycolysis doesn't require oxygen, right? And thus, that's probably something where you can get a little bit of ATP from it, as we studied in Bio 1. So this is a mitosome, reduced mitochondria, no functional ETC, thus can't use oxygen. Another thing to understand about the Diplo Monads is that they actually lack plastids. And this is going to be important because most protists have plastids. Most protists are going to be utilizing their plastids for photosynthetic processes. Instead, Diplo Monads classify as those protists that don't have plastids, so it's important to remember that they lack plastids. In addition, their name, this diplo part of their name, comes from the fact that they have two equal-sized nuclei. So that's a pretty cool fact to know about them, just to remember their name, two equal-sized nuclei, thus the name Diplo Monads. And then finally, in terms of excavates, we haven't really mentioned the excavate nature of the Diplo Monads. Diplo Monads classify as part of the fact that many excavates, those that have these excavated feeding grooves, are parasitic. So they are part of these plus-minus relationships as we studied in ecology back in Bio 1. A good example of this, and you have to remember the examples, you have to remember the names, and you have to remember key characteristics. A good example of this is Giardia Intestinalis. This is a direct organism. This is a genus, Giardia, thus the capital G, and then I'm going to lowercase the species, Intestinalis. Intestinalis. So this is a protist that is classified as a diplomonad, which is an excavate, which is part of our protist overall group, and then that's part of our eukaryotes. Don't be able to go upwards and downwards down that chain that we just did. Giardia intestinalis, a little bit of backstory, it lives in the small intestine of humans, lives in small intestine of humans. And in terms of its clinical relevance, its medical relevance especially, this is otherwise known as the organism that causes backpackers' diarrhea. So this is often seen in people that are hiking a lot, and they will definitely understand that this is a parasitic relationship that causes this syndrome based off of this, that causes this result, this disease, based off of this intestinalis organism. So that's a good medical relevancy to understand about the Diplo Monads. In addition to the Diplo Monads, which are part of the excavates, which are type of protists, which are eukaryotes, we have to also understand the other subclades. The other one that I will mention in this video, last one for this video at least, are the parabasalids. Another name to remember. Lots of memorization in this lecture, but it's all important in terms of understanding the great diversity that we see in this protist group. Parabasalids, first and foremost, lack plastids. So doesn't that make sense to group two things, diplomonads and parabasalids, together because they converge on the fact that they lack plastids. So that's an important fact to note. These two lack plastids. Kind of weird to see in protists, as we'll see when we move forward and look at the protists that most of the protists do have these plastid structures. Again, key term to remember, characterized by, which I'll just abbreviate as CHAR, by, because I'm going to be writing it a lot. When we say characterized by, this is the defining characteristic, something you should remember for parabasalids. It's something called the hydrogenosomes. So whenever you think somes, these are bodies. These are probably going to be organelles within this cell structure. And that's exactly what it is. A hydrogenosome is an organelle. Within parabasalids, they also have reduced mitochondria. And their reduced mitochondria is for the exact same reason as diplomonads have a reduced mitochondria, and thus they lack a functional ETC. Lack functional ETC. So you should already be thinking, well, how do they get their energy? They certainly don't get their energy from aerobic pathways like anything involving oxygen, like the electron transport chain. What they do get their energy from is anaerobic pathways. So we'll write that down. Rely on anaerobic pathways for energy. So that's how they get their ATP, from without oxygen pathways. And then when they do this anaerobic respiration, they actually are going to release hydrogen gas. They release H2, which is hydrogen gas, and guess where they store it and release it from? the hydrogenosomes. That's the key characteristic to remember about parabasalids. Based off of their metabolism, their respiratory processes, they release H2. Okay, so it's very weird to see that in the entire living world, to see something that releases hydrogen gas. It's seen in the parabasalids, thus really exemplifying the great amount of diversity that we see by these organisms within the protist group of organisms. Finally, remember, we have to remember examples. We have to always go back and classify these things as real living organisms. And the example to understand here is Trichomonas vaginalis. And this is one of, again, medical relevancy, of clinical relevancy to us, because this is actually a common sexually transmitted parasite. And remember what we said about most excavates, they are parasites. They can use this feeding groove that we have here in their characterized by sentence right here and use it for parasitic processes, for plus-minus relationships. And this is a parasite that causes trichomonasis. So again, medical relevancy, clinical relevancy, something that is definitely important to remember in all of these study of protists. So that covers our first two subclades of Excavata. Diplo Monads, parabasalids are types of excavates. Excavates are types of protists, and protists are a type of eukaryotes. We'll continue this discussion in the next flowchart and conclude on one final excavate.